Namaskar. Welcome to our course on Introduction to Microorganisms. We are in chapter Bacteriology. In this module, we have finished Introduction, size and shape, prokaryotic or bacterial cell appendages, prokaryotic or bacterial growth, prokaryotic or bacterial reproduction. And we have finished one part of domain bacteria. We are into the second part of the three-part domain bacteria chapter. We are continuing with different types of bacteria according to the three domain classification of the living system. In this, the first organism we are studying is spirochetes. What are spirochetes? Spirochetes are helical heterotrophs, as can be seen with the image. Some of these are parasites, which includes Treponema pallidum, a causative agent of syphilis, and Borrelia burgdorferi, which causes Lyme disease. Spirochetes stain gram-negative. Spirochete, the name is derived from Greek, which means coiled hair. Spirochetes are extremely thin and can be very long. They are known to be from 0.5 to 500 micrometers in length, in comparison to E. coli, which is about 1 to 3 microns in length. Motility of spirochetes is by periplasmic flagella with axial fibrils or endoflagella. Their outer sheath encloses axial fibrils, and these axial fibrils originate from insertion pores at both poles of the cell. When classified, spirochetes belong to three human pathogens: Treponema, Leptospira, and Borrelia, while other spirochetes are found in water, sewage, and even in our mouth and genital tracts. Flagellar movement of spirochetes. They are strange when we are considering their structure, especially their flagellar structure. As can be seen here, the axial filament are within the sheath of the bacterium, and this sheath covers the whole bacterium and the flagella inside it. This sheath helps the bacterium in coiled type of movement, a motility which is typical for spirochetes, as can be seen here. This movement is the spiral movement of typical spirochete. Next are Filamentous Prokaryotes, mainly Actinomycetes, an important group. The Actinomycetes include Nocardia and related acid-fast organisms. These are also including Actinomycetes. Actines means ray, Mycetes means fungi. These were considered as ray fungi for their growth properties. They were initially thought to be fungi. Now we know them as prokaryotic bacteria. They are a diverse group of organisms. Some of them are also acid-fast. Actinomycetes are soil organisms. They are similar to both bacteria and fungi and have characteristics linking them to both the groups. Actinomycetes are often believed to be missing evolutionary link between bacteria and fungi, but they have many more characteristics in common with bacteria than with respect to fungi, including their condition of prokaryotic nucleus. When considered for their cellular similarity, we find actinomycetes like a mycelium as seen in fungi. They are also present as aerial mycelium like fungi. They form typical conidia like sporulation. They have flagella. Many of these actinomycetes show bacterial flagellum, and their prokaryotic cell structure is similar to bacteria. But overall, the colony morphology is like a fungi. As can be seen in this image, the colony morphology looks like a fungi, while the cellular structure, it also looks like a fungi. It is branched filamentous organism, while it is prokaryotic in nature. When seen for their growth in one of the important actinomycetes, that is Streptomyces, we can see the growth phase or the tip of the hypha or tip of the growing filamentous cellular body folds or curls and starts to form partitions. These partitions thicken and form the conidia or the spores on the basis of different types of spores, and the way they form the spores, they are considered to be different species of the organism. Actinobacteria are also high G+C content bacteria. They are gram-positive in nature. They include Mycobacteria, Coryneform, and Actinomycetes. It includes an important pathogenic organism like Mycobacterium, while Streptomyces and Actinomyces are known industrially very important. Streptomyces, Actinomycetes, Frankia and Nocardia are known to be good filamentous organisms, while Streptomyces and Actinomyces form conidiospores. Nocardia and Frankia form no conidiospores. While Frankia can fix nitrogen, Nocardia and non-mycelial Mycobacteria cause acid-fast surface. Coryneform is a non-acid-fast stain but causes diphtheria, while Propionibacterium is an industrially important organism producing propionic acid. They are often filamentous, and many of the organisms are known to produce antibiotics. Actinobacteria, which includes Coryneforms and Mycobacteria, Coryneform and propionic acid bacteria. Coryneform, as the name says, it is a club-shaped, irregular-shaped or V-shaped cell which is arranged in palisade. They are gram-positive, aerobic, non-motile, rod-shaped cells. They are extremely diverse group. Mycobacterium. It is a rod-shaped organism exhibiting acid-fast surface. Mycobacterium is one of those organisms which were identified by Robert Koch in early period of microbiology, when the microbiology as a subject was still nascent. These are called as acid-fast because they have high surface lipid content and cannot be easily stained by usual gram staining process. Next, key organisms, Streptomyces and Actinomyces within Actinobacteria. They are filamentous, gram-positive bacteria, produce mycelium analogous to mycelium of fungi. Over 500 species of Streptomyces are known. Streptomyces spores are called conidia. Most of these organisms are soil organisms, and that fresh earthy smell that we get during the first raindrops or during the water wetting of soil is because of geosmin, that is production of a chemical from actinomycete. These are strict aerobes that produce many extracellular enzymes and antibiotics. Streptomyces. 50% of all isolated Streptomyces are producing antibiotics, and over 500 distinct antibiotics are produced. Some produce more than one antibiotics. Their genome are typically quite large, and we still have to study Actinomycete more so that we can understand their ecology. Next group of domain bacteria are Cyanobacteria or blue-green alga. We have seen such mats of Cyanobacteria on fresh and marine waters. These mats are full of different types of cyanobacterial organisms. Cyanobacteria, as all prokaryotes, are lacking of organelles like nucleus, chloroplast, mitochondria, Golgi bodies, and even endoplasmic reticulum. They are known to be photosynthetic or also called as phototrophic. They form gas vesicles which helps these organisms to maintain buoyancy, to float on the surface of water, keep self in water column where there is light, that is, if and only if the organism can float on the surface, it can get more light than at the bottom of the water content. They form typical heterocysts, which are enlarged cells. These heterocysts are forming anoxic environment inside themselves, where nitrogen fixation can take place. We know nitrogenase enzyme, which fixes atmospheric N2 into ammonia, is sensitive to oxygen, and hence the heterocyst availability of anoxic condition helps nitrogenase activity. Some of the pigments that Cyanobacteria have is chlorophyll, mainly chlorophyll A, sometimes chlorophyll A and B. phycobilins, which includes phycoerythrin, phycocyanin and allophycocyanin. Carotenoids, which are a variety. Some are specific, others are in algae and plants also. Cyanobacteria are known to have storage products, which are cyanophycean starch made up of 1,4-glucan, cyanophycean grains, which are nitrogen reserve, a copolymer of two amino acids, aspergine and arginine. They also show volutin grains or polyphosphate granules. They also are known to store lipids as the storage products. When we say cyanobacteria are prokaryotic and they do not have cell membrane bound organelles, when we see their structure, we will come to know what that means. They form typical thylakoids with phycobilisomes. These phycobilisomes are on the thylakoid membrane, and phycobilisomes including phycoerythrin, phycocyanin and allophycocyanin are arranged on the membrane of the thylakoid. The cell structure is primitive, just like a usual prokaryotic cell, and each cell is composed of two parts, cell wall and a protoplast. The cell wall is composed of two layers, inner layer which is a thin film of peptidoglycan, and outer layer which is a sheet composed of peptic compounds. The protoplast consists of two parts, peripheral pigmented or colored region, while colorless inner region contains several chromatin granules, which are the DNA and are forming a primitive nucleoid. As can be seen in this image, the thylakoids are typical membranous invaginations within the cell or by the cell membrane, which form the photosynthetic apparatus in the organism. Cyanobacteria and their cell wall stay gram-negative as the peptidoglycan layer is less. On the basis of their morphology, they can be present as colonies or as single cells. They can be multicellular, like a trichome, or multiseriate trichome, where they are arranged in different forms. We can see a false branching within cyanobacteria, where in the presence of a heterocyst, the cells next to it, they turn and form a typical branch, or sometimes when a branch is needed for cellular growth into a particular groove in the environment, the mycelium or the filamentous cellular body of the cyanobacteria, it bends and breaks into typical folds, which actually, because of the presence of a linear cellular body, looks like a branch. Sometimes we also see true branching, but that is only seen in some representatives of order Strigonematales. When we say reproduction of cyanobacteria, they show either vegetative reproduction, that is by breaking or by growing of the cells, or by asexual reproduction, which includes by fission or by akinase. In these organisms, sexual reproduction is not known. We don't know. They might have it, but we have not yet seen it. So we don't know. As can be seen in this image, the vegetative reproduction is by binary fission and fragmentation. Sometimes hormogonia, which are only in some filamentous forms, are important vegetative reproductive organs. They separate the filamentous organism into 2 or 3 different individuals that grow separately due to these separation discs. Akinase. As seen in Anabaena, these are resting cells with thick cell walls and enriched with storage products. These sometimes can be considered as the spores. Sometimes there are actual spores, endospores and exospores, but these are only in some unicellular forms which can actually produce these. We have not seen this sexual reproduction, but by viral transduction may happen, as we will discuss about this in our future modules. Heterocysts, as we discussed, are nitrogen fixation centers. Importance of cyanobacteria: they fix nitrogen, they can be used as food. Sometimes they are known to pollute the water source, while some of them can produce toxins, hence it is called as a pollution. High concentration may cause fish toxicity and other microorganisms may also grow with them. Now we come to stromatolites. Stromatolites are one of the primitive cyanobacteria. These are the oldest living organisms that we can see today. In a way, they are living fossils. They are considered to be the first organism evolved on primitive Earth with the ability to photosynthetically harvest the solar energy. They are mainly cyanobacteria, and their fossil record also is included 2.8 billion years ago. Wherever they are present, they are dominant, as there are no herbivores are present. Here, domain bacteria section 2 completes. Let us continue with domain bacteria section 3.